Hello, everyone. Today we are going to walk through doing an entire calculation set for a one-way ANOVA, and I'm going to do this with an example. And this example is data from an experiment where the effect of salinity level on magnesium ion concentration in the hemolymph of estuarine lipids is what we're interested in finding out. So this picture on the right-hand side shows what a limpet looks like. If you don't know that word or name, these are these little sort of oval shaped shells, kind of peaky at the top, and you find them on rocks at the seashore, and they stick on really tight. You might have heard of the phrase stick on like a limpet, or you might not have, but it was used in my family. All right, so in an estuary, there's changes in fresh and salt water throughout the day so that these organisms need to, oh, they're a mollusk. So these organisms need to be able to regulate their different ion concentrations. And hemolymph is the word that's used for organisms with an open circulatory system like mollusks have instead of blood. So basically, the purpose of this experiment is to see how um, these small snail-like animals are able to regulate their magnesium ion concentrations. All right, so the salinity levels, those are different concentrations or amounts, and about 33 parts per thousand of seawater is full seawater. So these are 15, 20, 25, and 30 parts per thousand of seawater. So we had collected a small number of limpets, and they were put into tanks that had water put into them that was set at these different levels of salinity. So they were exposed to that treatment for 24 hours, and then a tiny blood sample was drawn from the animal, and its magnesium ion concentration was measured in a magical instrument that is capable of measuring such a thing. All right. So this is suitable for an ANOVA style experiment because we have 4 different treatment levels, and we want to look at the effect of this categorical levels of salinity. We didn't do every single salinity there is. We picked 4 separate ones. We've got independent sampled individuals, so different lipids in each of the 4 treatments. And we're going to look at the data first. So if you're working along with this example, take a moment here and pause the video so you can copy down all of the data values. Now, this is a really small sample set, and that's a little unrealistic because we want to have a sample that's bigger than this typically, although this theoretically meets the requirements of ANOVA because we have an even sample size of 5 or more across our 4 different treatments, right? So the calculations for this are have a lot of steps. So I didn't want to give you too big of a data set or we'd be here all day. All right, so we've got first individual in the 15 level of salinity, and we measured its magnesium ion concentration as 114. All right, so each of these measurements in the table represent a measurement on a different individual limpet. All right, and they range from 110 to 120. All right, well, we're getting ready to do an ANOVA. There are some things that we like to use as summary statistics and things that we need to identify from the experimental design in order to kind of set up how we're going to do the analysis. Some of these things software is going to do for you, but it's good if you know the where those numbers are coming from. All right, so first for our hypothesis test, we're going to write the null hypothesis, which is that the true means of each of the four treatments are equal to one another. So our H sub 0 is mu sub 15 equals mu sub 20 equals mu sub 25 equals mu sub 30. Right, so another way to say this hypothesis in words is that all the means are equal. Right, if all the means are equal, then there's no effect of salinity level on the magnesium ion concentration. Now, our alternate hypothesis, I've written it out this way. Mu sub 15 does not equal mu sub 20, does not equal mu sub 25, does not equal mu sub 30. You can also write it as, and some statisticians think it's more correct to write it as not all means are equal. Because if we reject the null hypothesis that all the means are equal, right, it doesn't necessarily mean that every single mean is different from every single other mean. All right. So I like to write it out so that it matches the null hypothesis. I think that makes it easy to understand in some ways, but you might see written in other resources, it written as not all means are equal. All right, so what we want to do as we're getting started to wrap our head around our experimental design, we want to identify capital I, which is going to be the number of different treatments that we have, capital N, the total number of individuals in the experiment, and we're going to find it by looking at the little n sub I, which is the sample size of each of the treatments. All right, they can be equal to one another, but they don't have to be. All right, for exploratory data analysis, we're going to calculate the means and the standard deviations of the groups. We don't actually need to calculate these in order to find the sums of squares to calculate the ANOVA table, but they're nice to graph so that you can see what the pattern of the data look like. And then we're going to need the grand mean. All right, so we'll have the means of each of these different groups, and then we're going to take the mean of the data set overall. And you can find that by finding the mean of the means, and it's often used with the notation x double bar. So that's how you'd say that. X bar is our mean, x double bar is the mean of the means, or the grand mean. All right, so we're going to want to make our graph of the means showing the error bars. And in this figure, in this slide, I've repeated the data here so you can see that the means of each of the groups are a little bit different from one another and calculated the standard deviations. All right, so if we're using Excel or we're just going to sketch a quick graph by hand like this one, we're going to put the salinity and the four different treatment levels on our x-axis. Right, so that's the independent variable, the one we, the scientists, have controlled. And we want to ask the question, what's the effect of salinity on our response variable, magnesium ion concentration? All right, so we want to put our numbers of the range our means are found across on our y-axis, and we're just going to plot the means as a dot or marker and show estimated or approximate in this hand sketch graph error bars around them. All right, so one of the assumptions of the ANOVA is that the largest standard deviation is not more than twice the smallest. All right, so that's a quick way of figuring out, are the standard deviations approximately equal? Now, these are really close to one another, but here's how we would do that. We're going to take this smallest one, 1.9, and we're going to multiply it times 2. That's going to give us 3.8. All right, we're going to look at our other standard deviations. None of them are bigger than 3.8. All right, so our standard deviations are close enough together. Now, when you're using software to do this test, it's going to do that test for you. and it will let you know, or it will automatically make a correction for it. All right, so let's find out our other summary statistics. We want to know capital I. That's the number of different treatments. And so we have 4. And our little n sub 1, or we can substitute in the actual names of our treatments here, or a code for the names of our treatments. So I could have written n sub 15, n sub 20, right, instead of just 1, 2, 3, 4. They're all the same as one another, and they all equal 5. Got 4 treatments times 5 individuals. That makes our capital N 20. And I calculated the X double bar just by taking the mean of the means here, and it comes out to 115.0. And our units for this concentration are, I think it's actually micrograms per deciliter. All right, so we've got our summary statistics calculated. We've got an idea of what our results are going to look like. Usually at this point, you want to take a look at the means and say, well, if any of the means are going to be different from one another, the ones that are most different from one another are the means from the 15 salinity and the 20 salinity. The 25 and the 30 aren't that different from one another. But we have to do the analysis in order to see which ones are indeed significantly different from one another. All right, so in order to complete this example, we're going to fill in an ANOVA table. So here's a review of what goes in the ANOVA table. We have the 3 different sources of variation: between groups, error, or within groups, and total. We're going to first find the sum of the squares, and we're going to do that by taking deviations in different ways. So I'm going to show you how that's done quickly in this example. And I encourage you to follow through and do the calculations because it's going to help you understand what we're actually finding when we do this whole ANOVA. But normally we're going to let software do that for us. Now, the notation in this particular example table is a little bit different than what I use. For example, they're just saying x bar here instead of x double bar, which is the notation I use. And these x bar sub j's, those are the group means. All right, so in each of these sums of squares, we're going to be taking a deviation. We're going to subtract either x bar from x or x double bar from x bar. And in this total one, we're going to be subtracting x double bar from little x. All right. Degrees of freedom, we're going to find that. Now, I use the capital letter I for my notation here instead of little k. So that's the number of groups, capital I minus 1, n minus I, and n minus 1. Right? Don't write with some kind of font in your handwritten notes where you can't distinguish between a capital I and a numeral 1. All right, and then we're going to find the mean squares by dividing the sum of squares by its degrees of freedom. We're going to have a value for the mean squares between groups. We're going to have a value for the mean squares within groups, also called error. And then we're going to find the F value by taking the MSB, in this example, divided by the MSE. All right, we're going to have to calculate or be given at least 2 of the sums of squares. We'll find the degrees of freedom from the experimental design. And once we have those, we calculate the mean squares and the F value. Then we'll go to the F distribution to find the P value. All right, so I set up the table. I'm calling this step 1. And we're going to do the components of the table, fill it in for this example. So for the degrees of freedom between groups, that's capital I minus 1. We have 4 groups, so degrees of freedom between is going to be 3. Capital N is 20. Capital I is 4. So we're going to have 20 minus 4, or 16, for degrees of freedom within. And then capital N minus 1 is 20 minus 1, or 19, right? Notice that they should add up, right? So between degrees of freedom between plus degrees of freedom within does actually equal the degrees of freedom total. All right, so it's really a total. All right, now we're going to have to calculate these sums of squares. All right, let's begin with the between. So the sum of squares between, excuse me, we're going to calculate it by taking little n for each group, multiplying it by the mean of the group mean, minus the grand mean. All right, so if you look back to the data table, you'll see that this is the group mean of the first group, 118 is the group mean of the second group, 14.2 is the group mean of the third group, and 115.4 is the group mean of the fourth group, the 30 parts per thousand. In each of these deviations, we've just subtracted the grand mean from it. All right. Then we're going to square those. And the order of operations here is that we're going to first do the subtraction, and then we're going to square. All right, don't square 115 first and then subtract it. Yeah, we're going to do the subtraction first, and then we're going to square. So you're going to get some negative numbers, you're going to get some positive numbers, but when you square them, they're going to all turn into positive numbers. So we should be adding up positive numbers here. Now we've got 5 times each of these deviation terms, so I've moved it outside. But if they're different from one another, then you just multiply it by them each across here as you go. All right. So you want to check with your calculator as you work through these example problem that you get the same number. So you should get, if you calculate out all the steps, you should get 82.8. Now, if you are not seeing where these numbers come from, look back at your data table and the means and the grand mean and make sure you're seeing where these numbers come from. Right, the sums of squares for the other 2 rows of our table are going to look similar but slightly different. Now, when we're ready to calculate the sum of squares within, there's a few more terms in our summation. We have a term in our summation for every single data point that's in the data set, not just one for each group. All right, so if you look carefully at these numbers on the left-hand side of these terms in parentheses, these are all of our data observations. These are the little X sub i's, and I have it laid out here on the page in the same order of columns and rows that you have the data in the data table to make it easy to see where these numbers come from. All right, so we've got every single observation here, right? And then each column is from the same treatment, and we're going to subtract its own group mean, right? So if you look at the data table, you see this number, and then down at the bottom, its group mean is 112.4. So our first term is going to be 114 minus 112.4 squared. Going down the column, we see we did the exact same thing with all of the data from that first group. And then across the rest of the page, we are switching out the mean for each column instead of the one we used in the first column. All right, so we've got 119 minus 118 squared, and we've got 112 minus 114.2 squared, and then 117 minus 115.2. All right. So we have all these terms. It's a little tedious to calculate all of these numbers. There's a lot of room for making mistakes as you are adding those up. And that's the reason why we're usually going to trust software in order to make these calculations for us. All right. But to practice understanding mathematical equation notation, I strongly recommend that you practice working through all of the calculations for this example. And you should get this value 69.2 as the answer for your sum of squares with it. All right, the sum of squares total is going to be each individual observation And if you look at the data table, you're going to see that each of these are the observations for each of the 5 different limpets from each treatment. And we're going to subtract the grand mean from each one. So in this case, we've got our entire data set arrayed here in each of these terms that we're going to, and we're going to have the exact same number, the grand mean. Is what we're going to subtract, right? So this sum of squares is going to be the total amount of variance within the entire data set because it's the difference between each observation and the grand mean, right? Again, pretty tedious to calculate if you have a large data set. It's not so bad if we have a fairly small one here. And once you do out all the calculations, you should get 152 as your result for the sum of squares total. All right, so let's fill in those sums of squares in our ANOVA table. We'll copy in 82.8 for the between, 69.2 for the within, and 152 for the total. Right, now going across the ANOVA table, the next column to fill in is the mean squares. Right, we've got the sums of squares. How do we find the mean squares? We divide by the degrees of freedom. All right, so let's do the next step. We're going to actually take the sum of squares between 82.8 and we're going to divide it by 3. So we see this pattern where we're now going across the rows, following across the rows, and we have all the information that we need. So we're going to get 27.6 for the mean squares between. You can do the same thing in the row for within, 69.2 divided by 16. That's going to give us 4.3. All right, now I've blacked out the box at the bottom of this row. We just stop. We don't do anything with there is no mean square total. All right, so it kind of has this stair step shape to the table when we get to the mean square and the F values. Right. So we're only going to have one value of F. We only have one value because it's a ratio between the mean square between and the mean square within. So we'll do that in our next calculation step. All right, so now we'll take sort of the table makes it look like it's actually a fraction. That's what we're doing, 27.6 divided by 4.3. That's going to give us 6.38. All right, so now we need to go to the F distribution table to find the p-value. All right, here's the section of the F distribution table. And we know what degrees of freedom we're going to use because it's in our, let's go back to that. It's in our ANOVA table, degrees of freedom. This is our numerator, 27.6, and its degrees of freedom is 3. So that's the numerator degrees of freedom. The denominator is for the mean square within, the degrees of freedom are 16. And so we're going to go to the F table that has 3 degrees of freedom in the numerator and 16 degrees of freedom in the denominator. All right, we've got our 3 degrees of freedom in the numerator, and I've got a black box here around. Now we have 15 degrees of freedom in the denominator. That is the closest value we have in our table. We don't have the exact value in the table. We're just going to have to use the closest one here. All right, so our value of F was 6.38. All right, so our value of F, we want to find where it is among these numbers, 2, 3, 4, 5, 9. Okay, so our value of F is in between 5.42 and 9.34 in our table. Make sure you find that. So we go over to the left-hand edge of the table and we find that our p-value is then going to be, right? And if you get lost in this table, remember to look back up at the top here and remind yourself that this column label is telling you these are the p-values. So the p-value for our result, for our value of F, is going to be between 0.01 and 0.0. between 0.01 and 0.001. All right, so I've put that in our ANOVA table here. Our P is going to be less than 0.01, but it's going to be greater than 0.001. All right, and the one that it's less than is more important. All right, so we're going to use an alpha of 0.05, basically for all of our initial hypothesis tests in our ANOVAs. All right, so we ask ourselves, just like we do in every hypothesis test, is our P less than our alpha? Is 0.01 less than 0.05? And the answer is yes. All right, so what are we going to do with that? We are going to reject the null hypothesis. All right, look back at our null hypothesis if you need to. The null hypothesis was that all of the means are equal, so we have rejected that. All right, but all we know is that at least two of the means out of the four means are not equal to each other, and we don't know which. All right, so we have to do some work. If we reject the null hypothesis for ANOVA, we are not finished. And now we want to know which of those means are going to be different enough from one another that we'll say they're significantly different. Okay, so these are called follow-up tests. Sometimes they're called post hoc comparisons. That'll be written POST-HOC. It just means after the fact. All right. So we have to do pairwise comparisons of every possible combination of the groups. All right. So just using the codes for our different treatments, we have to compare the mean for 15 to the mean for 20. We have to compare the mean for 15 to 25, for 15 to 30, and being systematic, just like we did when writing out sample spaces way back in probability math. And then the next one that we haven't written down yet is going to be 20 compared to 25, 20 compared to 30, and finally 25 to 30. All right, we don't have to also do the reverse. If we did 30 compared to 25, we get the same answer. All right, so it's not like probability in that regard. All right, so our ANOVA is an extension of the two independent samples t-tests. So when we're only comparing two means to each other at a time, we're going to do two independent samples t-tests on each of our pairs of means. All right, so we have four groups in our experiment. Our big I is 4, and that means we're going to have 6 total follow-up comparison tests we need to do. Right, bigger experiments, this gets pretty clunky. And there are ways that statisticians decide on what comparisons they want to make ahead of time and simplify the process. All right, we're just going to do this very straightforward way. All right, I'm not going to do all these t-tests one by one for you. I'm actually going to give you the p-values here so you know what the results from those tests were. All right, so we've got the p-values in here, but because we're doing 6 different tests, we need to do the step where we adjust the alpha level. And this is called, this is the simplest way to do it. It's called the Bonferroni alpha level adjustment. And we're going to take our overall ANOVA alpha level, and we're going to divide it by the number of comparisons that we made. So this number 6 of how many little t-tests we did. We're going to use that. All right, so we're going to take 0.05, we're going to divide by 6, and that gives us 0.0083. So that's a pretty strict alpha level that we're going to use here. All right, so once we get our p-values, we aren't just going to run here and say all the ones less than 0.05, we're going to reject. We actually have to compare them to this 0.0083. We're only going to reject the null hypothesis with p is less than 0.0083. So if we come over here and we look at all of these p-values, 0.002 is the only one that's small enough for us to reject. All right, so it's also handy to write a little table for yourself of which of the null hypotheses you're going to reject. All right, we're not quite done. We want to summarize our results, tell everyone what we found in our analysis, and we're going to do that in words. So I've put the graph up here, which really helps looking at the overall results. Only um, difference we found that was statistically significant was between the 15 and the 20. So that's the one we should find. The one that says the biggest actual numerical difference between it is the one we should find is a significant result. But the ones that 15 wasn't big enough, wasn't different enough from the 25 or the 30 to reject those nulls. The 20 wasn't big enough compared to the 25 or the 30. So the proper way to write your conclusion is in two parts. The first one about the overall ANOVA hypothesis. The overall ANOVA hypothesis that all means are equal was rejected. Follow-up comparisons revealed that mean hemolymph magnesium ion concentration was statistically significantly different between the 15 and 20 parts per thousand treatments, but not significantly different between any of the other pairwise comparisons. It sounds a little clunky, but you need to specifically state for each pair of means uh, what the result was. Don't just say these were significantly different and leave us hanging on the rest of them. All right. So you can sort of summarize the overall results if it's something that can be summarized, not all the others, right? Okay, I hope this helped you understand how to do calculations to find the results of an ANOVA hypothesis test.